Hello, in this video I'm going to talk about nucleic acids. So along with proteins, lipids, and carbohydrates, nucleic acids are another one of the important biological molecules that we find inside of cells. In terms of their structure, like the other organic molecules, they contain a carbon skeleton. And attached to this carbon skeleton are other things like monosaccharides. So actually we have a sugar attached in here, and there's lots of nitrogen and lots of phosphate groups. And we'll talk more about the structure of nucleic acids in a little bit. I did want to mention that like the other biological molecules, we have a monomer and a polymer here. So the single unit, the monomer of a nucleic acid, is called the nucleotide. And the polymer is actually the same as the group name, it's nucleic acid. Nucleic acids have many different functions in the human body. DNA and RNA are two examples of nucleic acids that you may have heard of, and these are both important for things like gene expression and heredity. So DNA is our molecule of heredity, and it contains the information to make proteins. And when we read the information in DNA to make proteins, we are said to be doing gene expression. So obviously you need DNA for this process, but you also need RNA for gene expression. The process of reading the information in DNA and making a protein requires RNA as a helper. So both DNA and RNA are needed for gene expression. You might have also heard of another nucleic acid called ATP. So I mentioned ATP in my carbohydrate video because I said carbohydrates are important for energy, but cells can't use carbohydrate energy directly. Instead, what cells do is they convert carbohydrate energy into the energy of ATP. And ATP is the energy currency that's actively used by our cells. ATP is classified as a nucleic acid. In terms of nutrient sources of nucleic acids, this is an area that we're less familiar with, just because when you look at a nutrition facts panel, you see things like proteins and carbohydrates and fats, but you don't see nucleic acids. So nucleic acids are in the foods that we eat, but they're not there at a level that's very significant nutritionally, so it's not included in a nutrition facts panel. But most of the foods that you're eating contain nucleic acids, like fruit, for example. So looking at the structure of nucleic acids in more detail, again, we have that polymer-monomer pattern here. The polymer is nucleic acid, and the monomer is called the nucleotide. So we take a whole bunch of nucleotides and connect them one to another to get a nucleic acid. So let's take a closer look at the structure of a nucleotide. So the nucleotide has three basic parts to it. Of course, it has a carbon skeleton to it, but as a part of that carbon skeleton, we have a sugar, and attached to that is a phosphate group, and also attached to that sugar is a nitrogenous base, which is just a carbon skeleton base with lots of nitrogen in it. So in the picture down at the bottom, we see sort of a cartoony version of a nucleotide structure, with the three main parts shown in red, yellow, and blue: the phosphate group, the sugar, and the nitrogenous base. So every nucleotide has at least one phosphate group. Some have more than that. ATP stands for adenosine triphosphate. ATP happens to have three phosphate groups. Okay. All nucleotides have a sugar base to them. And the sugar can be one of two kinds. It can either be a ribose sugar, which we find in both RNA and ATP, or it can be deoxyribose sugar, which is found in DNA. Ribose and deoxyribose sugar are almost identical. The only difference is whether or not there's a hydrogen or a hydroxyl group in one single position. In addition to the phosphate group and the sugar, all nucleotides also have a nitrogenous base. And the nitrogenous bases come in 5 different varieties. They're best known by the letters A, G, T, C, and U. On the right-hand side, bottom of the screen, we can see the 5 different structures representing our 5 different nitrogenous bases. You can see how they all have a carbon skeleton to them, but they all also contain nitrogen, hence the term nitrogenous base. The nitrogenous bases A, T, and C, and G are only found in DNA, while A, G, U, and C are only found in RNA. So the difference between DNA and RNA in terms of nitrogenous bases is the T versus the U. So now I want to look in more detail at nucleic acids, the polymers in this particular category. So the two important polymers we want to look at are DNA and RNA. And they're both important in the process called gene expression. So you've probably heard of genes before. Genes carry the code for making you, right? Well, genes are made from DNA. When we do the process of gene expression, we're reading the information in the DNA to make a protein. And in order to do this, RNA is needed. So basically what happens is we make an RNA copy of a gene and then we use that for making a protein. So let's take a look at DNA first in more detail. So in terms of its structure, DNA is composed of nucleotides, many nucleotides strung together. And all of these nucleotides have the deoxyribose sugar, and then they use one of the four nitrogenous bases, A, T, G, and C. So DNA is actually found in a structure called a double helix. And what that means is that we have two strands of nucleotides side by side, and then they're twisted in a helix, kind of like two strings of pearls side by side, twisted. On the left-hand side of the screen, you can see a flat version of this, but down towards the bottom, you can see the helical nature of DNA. The nucleotides within one polynucleotide strand, so here's one polynucleotide strand, these nucleotides are held together by covalent bonds, right? But the nucleotides between the two strands, such as between nucleotide 1 and 5, those are held together by hydrogen bonds, right? So if you think of DNA like a ladder, right? The sides of the ladder are the covalent bonds, but the steps of the ladder are hydrogen bonds. And it just so happens that the nitrogenous base A always hydrogen bonds with T, and then the base G always hydrogen bonds with C. And we call that complementary base pairing. In terms of function, as I said, DNA is important for gene expression, right? The sequence of the A's and T's and G's and C's in DNA is very important. Just like the sequence of letters in words is important. If the sequence of letters in a word is messed up, you won't understand what the story is trying to say. So if the sequence of the A's and T's and G's and C's in DNA is messed up, you won't have the right code for making the right protein. So the sequence of A's and T's and G's and C's tells the cell how to make protein, and we call this process gene expression. A gene is a specific sequence of A's, T's, G's, and C's that code for a specific protein. For example, let's say the code TACGGCGTTA codes for the hemoglobin protein, but then the code CACTCATCTCG codes for the protein insulin. In reality, these codes are much longer than that, but I'm just trying to give you a simplified example. So let's take a closer look at RNA structure and function. So in terms of its structure, RNA, unlike DNA, is single-stranded, not double-stranded. And instead of the nitrogenous base T, it contains the nitrogenous base U. And instead of the deoxyribose sugar, it contains ribose sugar. In terms of its function, RNA is a helper in the process of gene expression. So DNA carries the code to make a protein. But in order to complete this process of reading the code in DNA and making the protein, we need RNA to help the process along. The last nucleotide I want to talk about is ATP. So ATP is classified as a nucleic acid, but it's not a polymer, it's a monomer. It exists as a single nucleotide, unlike DNA and RNA, which are polynucleotides, if you will. But just like any other nucleotide, ATP contains a sugar, a nitrogenous base, and it has 3 phosphate groups instead of 1. In terms of its function, the bonds between the phosphate groups contain transferable energy that can be used by cells. So a phosphate from an ATP can easily be transferred to another molecule, and when we transfer that phosphate, we're essentially transferring energy. So we can't use glucose energy directly in cells. Instead, our cells have to convert the glucose energy into ATP energy, right? Much like gold bars are worth something, right? They're worth a monetary value, but you can't go to Starbucks and pay for your coffee with a gold bar. You have to convert your gold bars into dollar bills. Your cells don't have the enzymatic machinery. They don't have the proper setup to use glucose as a direct energy source. But they do have the machinery to take glucose and convert it into ATP energy. And then they do have the machinery to use the ATP as an energy source.